So as promised, now we are back to the topic on how polypeptides or proteins are made. Right? Remember we discussed DNA quite a bit, and we talked about DNA coding for proteins or polypeptides that lead to proteins. Right? So now we're going to discuss and fill in some of the steps in between. Okay, we talked about it briefly earlier, but here are more details. Okay, so polypeptides are made through a two-step process. Okay, remember we have DNA, then we have one step over here, RNA, then another step over here to get our protein. Okay, and the different steps have a very specific name. Okay, from DNA to RNA, the step is called transcription. Okay, so and then from RNA to protein, it is called translation. Okay, so if you can't remember, think of it like in two steps, right? Okay, so let's say you have a speech, someone talking, right? So someone's talking, let's say in a court or something like that, right? So you have someone that's transcribing, right? Typing it out, right? So that's the first step. And then you have someone that, let's say, is in a foreign language. Then you need someone to translate it into another language, right? So you transcribe first, then you translate. Okay, so from DNA, you transcribe it to RNA, and from RNA, you translate it to protein. Okay, so this occurs in different parts of the cell as well. Okay, so transcription occurs in the nucleus, right? Whereas translation occurs in the cytoplasm. Okay, it occurs in two different places of the cell. Okay, so basically transcription, if we have it in nice language, right? So transcription is the process by which the DNA template is used to make a single-stranded molecule called messenger RNA, okay, or mRNA. And this mRNA is single-stranded, right? And this has the base sequence that's complementary to that of the DNA template. Okay, so what happens next? This mRNA, right, it carries the message from this DNA. Remember, it's the codon, the triplet code, right? It goes out of the nucleus to the cytoplasm. Okay, and you get your protein being made. Translation. Okay, remember we've been mentioning this a few times that RNA contains uracil instead of thymine. Okay, so that's the only difference. The other, there's 4 bases, right? The other 3 are the same. The only difference is there's no T, there's no thymine in DNA, but instead it's a U, okay, or for uracil. Okay, so we talk about transcription, right? Translation is the second half, right? Translation is basically this sequence of RNA or mRNA is used to make a polypeptide. Okay, over here. All right, so some of you may be a bit confused by some of these terms. Okay, so we've added this here so that you can kind of remember. Okay, so replication is DNA replicating itself, so duplicating itself, right? So this is just getting more DNA, more of the same molecule. Where do we see this? Mitosis. Okay, that's this guy over here. Okay, what we're talking about over here is to get proteins. Okay, so replication is over here to get more DNA, but you need transcription to get RNA, and you need translation to get polypeptides or proteins. Okay? So basically DNA directs RNA synthesis, and through RNA it controls protein synthesis. Right? Remember the stepwise process. And RNA therefore is an intermediate in this whole step. Okay? So this flow, sometimes you may see this is called the central dogma of molecular biology or gene expression.